Greetings, I'm Bill Mobley, and this is another in our series of Neurosciences Connections. I have with me today two really distinguished scientists from our department who are going to talk about solving a huge problem, and that is a problem we refer to as the translation gap. You may know, very likely in your own experience, that very many millions of Americans suffer from neurological disorders. About 600 different disorders have been named, and each of them exacts a very substantial price on the well-being of patients and their families. So what's to do now? What can we say about these disorders? What we can say is that we can treat a number of them. We can cure none of them. And we have a big gap between then the wonderful work that goes on in the laboratory and the terrific work that goes on in the clinic. So we have great teams of people doing the science and doing the medicine. What prevents the one from moving their ideas to the other? What prevents great science from being great medicine? We have to solve this problem if we're going to help our patients. So today I have with me Mark Tuszynski and Steve Wagner. They've done a terrific job at creating an idea, a brand new idea here at UCSD, the Translational Neurosciences Institute. I'm going to ask each of them to talk about their lives, their careers, their interests, their goals, and how they fit into this new Translational Neuroscience Institute, and how we hope that this will make a big difference in what we have to do in the future to do our research, to translate great science into great medicine. So Mark, welcome. Steve, welcome. Glad you're here. Mark, tell us a little bit about yourself and about the work you do and your hopes and goals for the TNI.
Okay, well, it's a pleasure to be here. Let me begin by saying that we live in a golden era of neuroscience when the potential to really impact the human condition is remarkable. And in the current era, we really have the potential to take some of the great advances that have come through basic neurosciences in the last 30 years and really apply them to the human condition. Bill, you began by saying that we haven't seen this impact the human clinical arena yet. And we have, but to a very limited extent. And I think that it's most accurate to say that the need remains enormous, and the need persists for some of the most common and tragic neurodegenerative disorders and traumatic illnesses and genetic illnesses that affect many people. But at the same time, there has been the first translation into the human arena of therapies that have been remarkably successful and really highlight the potential of what could come. So why don't we begin by talking about some of those successes and then go on to talk about what some of the great need is. So the first success I would point to is in the realm of multiple sclerosis. This used to be a disease that we would poorly manage with steroids. Multiple sclerosis affects up to 500,000 people in the country. As you know, it's a disorder in which people lose the insulation around nerves of the nervous system, and that occurs progressively over time with flare-ups and that sort of thing. But people really, until about 20 years ago, faced no real therapies that could slow the disease. And beginning about 20 years ago, based on preclinical science that showed the role of the immune system in contributing to multiple sclerosis, a series of drugs became available that today constitute effective therapies for slowing the course of the disease. And the current prediction is that the course of the disease is slowed by about 1/3 to up to 1/2 with the latest generation of drugs. And that's a success. It's not a cure of the disease, so I guess you did rightly say we haven't cured diseases, but it's a significant treatment that has improved the lives of countless people with multiple sclerosis. So that's an example that we can use to gain some inspiration that if we can do this sort of thing in a variety of other diseases, we could have a positive impact. And let me just cite a second positive example, and that's the use of tissue plasminogen activator to treat acute stroke. This is, in a sense, is a miracle drug. Very few people get it because people have to come in within 3 hours of their stroke to be a candidate. But this is a drug that can literally dissolve away clots and take somebody who is paralyzed on one side to free movement of that side and recovery. And that's based on understanding the biology of how clots form and good chemical drug designers coming up with fits of novel drugs to actually have this mechanism of action in the patient, and then recombinant DNA technology actually producing this drug and bringing it to people with disease. So those are two examples in a field of about 600 that still need treatment. So the need remains enormous, but the potential is also enormous.
So Mark, you mentioned that these are signs of hope, that in fact it is possible to translate. what are the clues from those two examples that teach us that it should be possible for the other 598 disorders?
Well, I think the clues are doing good science and learning and understanding disease mechanism, and from that getting new clues into how to translate disease. And then the very difficult job of actually then translating this to the human arena, doing careful, well thought out clinical trials and getting that signal that, yeah, you do have something here that will be effective to treat disease. So, you know, in the last 20 and 30 years, this has been the realm of what we call molecular biology, where we've made basic discoveries about genes, about the proteins that those genes make, about how cells function and die or survive, and what kind of biological mechanisms we can recruit to stimulate cell survival, to prevent cell degeneration, to help cells form new connections, to protect cells from mechanisms that ordinarily cause disease. And there are today dozens of potential candidates out there that could achieve these things. But there is this gap, as you said, in translating all of that outstanding science to the human clinical arena. And why is that gap there? And in part it's there because we often lack a mechanism to think about how we can practically then take these very promising designs developed in many cases by basic PhD type scientists and change them into a clinically practical formulation that we can move on to the clinic. And that is a gap that we hope to fill here at UCSD through the formation of the Translational Neuroscience Institute, or TNI as we like to call it. And this is a new institute in the School of Medicine that will take a broad approach in bringing in new technology to basic scientists and clinicians that can help us further discover new mechanisms of disease. We'll bring in experts who are experts in drug design and combinatorial chemistry and protein synthesis to develop drugs for those targets developed by new insights into mechanisms, and then have another component of experts in clinical translation who will assist with thinking about how you design the clinical trials, the regulatory components you need to address, you know, very important but kind of mundane practical details. We'll provide that as a resource, and then implement in outstanding clinical arenas the testing of these therapies.
So it has all the pieces that you need in that puzzle. And the question now, I guess, is how we go forward to make those puzzle pieces fit together very well and for each of them to be robust. Now, one of them is clearly the drug discovery piece. And academics are not always great at discovering drugs, but we have Steve here, and Steve's been very involved in exactly that sort of thing. So Steve, talk to us a little bit about your background and what you see for your role in the TNI.
Sure. Well, I've spent about the last 25 years really working on identifying small molecule therapeutics for neurodegenerative diseases with a real emphasis on Alzheimer's. And as Mark mentioned, the real key to progress in any disorder is really understanding the mechanism. And so the better part of those 25 years have been really coming up with what I call a validated therapeutic rationale. And that's why I think those components of the TNI are every bit as important, if not more so, than the more practical aspects of drug discovery, which we've all heard about: high throughput screening, chemoinformatics, medicinal chemistry, structure-activity relations. And ultimately, I've been involved with all phases of what I call the target and drug discovery process, which involves identifying a molecular mechanism, and we've used genetics very much to our advantage. As Mark mentioned, this is really a key time, especially in neurosciences, where we're now learning to using bioinformatics, so to speak, to really understand how a gene or a mutation in a gene translates into a disease. And we're understanding the pathways much better. And once you really get a handle on a molecular mechanism that actually can cause a disease process, it gives someone with my background a real chance to go really try to identify tools to be able to intervene in that process. But without a real sound genetic mechanism, you're really just throwing darts on a dartboard. And that's why, as Mark mentioned, that it's very key that we take a very sound approach to rationalizing approaches, targets, mechanisms that we can exploit and use all the tools that are available in this community that expand from state-of-the-art high-throughput screening. There's now worldwide access to a host of different small molecule libraries that we can use to identify very specific, safe, and hopefully ultimately efficacious molecules that we first test in animals. We actually start back with cell-based models, very simple models that we ultimately translate, and the translation becomes more complicated as we get into the clinic, obviously. But so again, I think I'm very excited about this.
You sound hopeful. You sound like you think this is really doable. Both of you sound like you think it's doable.
So I'd like to point out that one of the things that's different about the TNI that we're trying to create is to bring in people like Steve to this whole effort. So Steve has over 20 years of experience in the industry world in attempting to develop and bring drugs to clinics, and you've done that, and you've brought drugs through clinical trials. And it's exactly that diverse expertise and background that can make this a success and really lead to the identification of effective therapies in humans. Let me come back to your original question, which is, what's different about this? Why does that gap exist? And part of the reason is that we haven't had an integration of the basic scientists with the people who have actually been out there doing the clinical trials and involved in every step in between in trying to develop new drugs. And having a diverse set of people with expertise in all of those areas together, talking together, discovering drugs together, thinking about how you implement this in the clinical arena, that could be key here to really breaking through and showing success. And Steve is a great example of that sort of expertise.
And we want that to be true. This integration seems to me so important. It seems to me that you can't do this just because you're a smart scientist or just because you're a caring doctor. You need this interface with industry, and at least with the technologies that industry has developed so well, to turn great science into great medicine. And so we're hopeful, of course, that that's going to happen right here under your leadership, Mark, and we're eager to see that become a fruitful endeavor. Early targets, early thoughts about disorders that you're most interested in looking at?
Well, the most common human neurodegenerative disorder is Alzheimer's disease. That affects 5 million people, and there, tragically, we lack any sort of drug that modifies the course of the disease. That's been identified broadly as a huge need. And there are many drug candidates that have the potential to do that. I've been involved personally in growth factor trials in Alzheimer's disease. The biology of these growth factors is remarkably potent in the animal models. But showing that efficacy in the human arena has been far more challenging. And part of the challenge there has been, how do you deliver these proteins into the brain over a sustained period of time? And again, bringing together a variety of technologies, we have the potential to do that. So that's one topic that's high on the list.
Very exciting, yeah.
So is Parkinson's disease, the second most common neurodegenerative disorder, affects 500,000 to 1 million people. And there are similar candidate drugs, growth factors, gene manipulations that can enter into that arena. A good example of the potential that has yet to sort of show its value in the human arena are things like gene therapy, where we have the potential to replace therapeutic genes in the brain through a single administration, providing lifelong delivery of a drug. It's a new way of thinking about drug delivery. It's a new mechanism for treating childhood diseases like inborn errors of metabolism, where again, one can make an early intervention, replace a deficient gene, and perhaps cure the disease. These were pie-in-the-sky dreams 30 years ago. These days, the tools have really progressed to the point that there is realistic potential to effectively treat these disorders. But you really need to integrate a set of scientists and instrumentationalists and neurosurgeons. We need collaboration among the whole set of people if this potential therapy is really going to show its ability to impact the human condition.
Steve, you're new here.
Right.
You've got a chance to sample the community. What's the prospect for real integration?
Well, I'm new to UCSD, but I'm not new to La Jolla. And I think of all the places in the world where, if one were to want to start and to build an institute like this has got to be the #1 choice because of, as Mark and you both mentioned, La Jolla has every component that we've all talked about, from the very basic discoveries to identifying the disease, finding the compounds, and actually the hard part, which is really the most challenging, and that's translating what we've had reasonable amount of success with in identifying efficacious molecules in what appear to be very reasonably well thought out animal models for these very devastating disorders, and then taking those into the clinical arena. So La Jolla is one of those places in the world that has all of the pieces of the puzzle, and I think it's a very exciting opportunity to be now a part of this, and I'm very excited to get going, like you mentioned earlier.
We're excited. We're all excited. The TNI has enormous potential. I believe in the potential, and I think you're going to see amazing things come out of UCSD and its TNI as we bond together with other folks at the university and La Jolla more generally to turn great science into great medicine. Thanks for watching.